and then duplicated chromosomes are inherited by newly born daughter cells. So this process is called chromosome segregation, and then we are very interested, how this is regulated, and then especially, what is the fundamental mechanism regulating this process. Bi-orientation is very crucial. It's at the heart of chromosome segregation mechanism, because if sister chromatids doesn't attach to mitotic spindle from the both poles, then chromosome missegregation will result. So therefore, this is very important basic mechanism ensuring chromosome segregation and then inheritance of a complete set of chromosomes. So it's an important topic in basic cell biology. But it's also important in medicine because when it goes wrong, often leads to cancer and then also various genetic diseases.
Bi-orientation can only occur during mitosis, when the nuclear envelope is broken down. This allows the microtubules to extend from the spindle poles and interact within prometaphase, referred to as open mitosis. In yeast, the nuclear envelope is not broken down and can be referred to as closed mitosis. A single microtubule elongates from a spindle pole to join to the lateral side of the kinetochore. It attaches to the lateral side due to the larger surface area. The microtubule lattice helps to provide an efficient first encounter with kinetochores. The NDC-AT complex is an outer kinetochore component, which is the main point of connection to the microtubule. The NDC-AT-NUF2 globular domain directly interacts with the microtubule lattice, without showing preference for microtubule ends. The NDC-AT and NUF polypeptides are folded as an actin binding site known as the CH domain. Positively charged residues in the CH domains are important for microtubule lattice binding. The NDC-AT complex is associated with other kinetochore components, KNL1 and Mis12 complex. This interacting protein set, called the KMN network, can actually cause higher affinity for microtubules than the NDC-AT complex can alone. Kinetochores are then transported towards the spindle in a sliding manner along the microtubule. This is stimulated by -end directed motor proteins, such as dynon, causing rapid kinetochore sliding in vertebrate cells, and CAR3 causing low intermittent sliding in bud and yeast. Microtubules maintain their dynamic nature of elongation and contraction at their plus ends distal to the spindle pole, even though kinetochores are being transported along the lattice. The DAM1 complex is crucial for chromosome motion in prometaphase, taking advantage of microtubule depolymerization, creating a pulling force for the kinetochore. DAM1 forms a ring that will be pushed poleward by protofilament curling, making it an ideal device for end-on pulling kinetochores. DAM1 complex particularly might be more crucial for organisms like bud and yeast due to only having a single kinetochore attached to a single microtubule in metaphase. DAM1 complex may not be required in metazoan cells because they use several microtubules per kinetochore, and in which components such as the KMN network may be sufficient for kinetochore association with the ends of the majority of microtubules, even if some microtubules may detach. When end-on attached kinetochores reach a spindle pole in vertebrate cells, they can slide along the surface of other microtubules that they are already attached to by orientated kinetochores, while maintaining their original end-on attachment. This sliding takes place away from the spindle pole and helps kinetochores to align in the middle of the metaphase spindle.
The B kinase has a central role in the orientation of chromosomes because all the B can kind of resolve erroneous or aberrant kinetochore microtubule attachment. And then that ensures bi-orientation for all chromosomes.
Aurora B kinase, or LPL1, is an evolutionary conserved serine/threonine protein kinase, crucial in promoting bi-orientation. Aurora B kinase localizes at kinetochores in prometaphase and metaphase. It promotes turnover of kinetochore microtubule attachments by phosphorylating kinetochore components. Crucial substrates include DAM1 complex in bud and yeast and the NDC-AT complex in mammalian cells. The Aurora B/LPL1 kinase facilitates bi-orientation by promoting orientation of the kinetochore spindle pole connections in a tension-dependent manner. Because syntelic attachment does not generate tension on kinetochore to pole connections, Aurora B/LPL1 promotes reorientation of these connections by phosphorylating kinetochore components. When an amphitelic attachment is established and tension is applied on kinetochore to pole connections, Aurora B/LPL1 stops promoting the reorientation, which causes preferential selection of the amphitelic attachment. Overall, the more we learn about the various components and mechanisms involved in bioorientation, the better understanding we will have on genetic diseases such as cancer, and hopefully provide the essential knowledge for future researchers to come.
On the other hand, if it's contaminated with media, that all